This is Week 5, Molecular Genetic Testing, Part 1. The learning objectives for this week are to discuss how DNA sequencing, southern blotting, and FISH tests are used to detect cancer, to explain and give examples of single-gene sequencing, targeted mutation analysis, and multi-gene panels, to discuss counseling and testing, options for cystic fibrosis and other single-gene disorders. Most cancers occur in people who do not have a strong family history of that specific cancer. They are sporadic. Some families have more of the same kind or related kinds of cancer than expected in the general population, with no specific pattern of inheritance, likely caused by a combination of genetic and environmental factors referred to as familial. Those in which the cancer is passed down generation through generation by inheriting mutated genes in the autosomal dominant mode of inheritance is referred to as hereditary. This only occurs in 5 to 10% of cancers. This slide is showing you some inherited predispositions to cancer and some of the genes that are affected. Determining which families have cancer related to an inherited gene mutation is important, since the cancer risks in hereditary cancer families are much higher than the general population. Following the published NCCN guidelines for increased surveillance, chemo prevention, and preventative surgeries, cancer risks have been reduced in many individuals with inherited cancer predispositions. As a genetic counselor, when doing a family history, you look for genetic red flags. You look for a family history of multiple affected relatives. You look for the condition in the less often affected sex, for example, breast cancer in a male. You look for an earlier age of onset of a disease than expected. You look for a disease in the absence of known risk factors. You look for multiple primary tumors in the same person. Look for bilateral disease. And non-cancer findings suggestive of a syndrome are very important to ascertain. You also look for ethnic predisposition as well as consanguinity. When taking that 3-generation pedigree, which is a very powerful tool, you will always show what is the type of cancer, the age of diagnosis, the ethnicity, and the results of any cancer testing in any relative, whatever you may find out. If an individual is positive for a genetic mutation, then family members may also carry the same mutation. First-degree relatives at 50% risk to have inherited the same mutation, second-degree relatives 25%, third-degree relatives 12.5%. A genetic counselor can help identify who in the family is at risk of having the mutation, who should be tested, and when they should be tested. It's very important to remember that some types of cancer may occur in several people in a family without being caused by a genetic mutation. There may be a genetic mutation in other members of your family, but you did not inherit it. So let's begin to look at tests to detect cancers and other mutations. We will look at first at the polymerase chain reaction PCR. In 1986, polymerase chain reaction became available to the scientific community. It revolutionized recombinant DNA methodology and accelerated the pace of biological research. In 1993, the Nobel Prize in Chemistry went to Kary Mullis, who developed this technique. PCR involves a rapid method of DNA cloning which eliminates the need to use host cells for cloning. It copies specific DNA sequences via in vitro reactions. It can amplify target DNA sequences present in very small quantities. PCR is a method of choice for many applications in molecular biology, human genetics, forensics, and evolution. PCR requires two primers. The primers are short single-stranded sequences, one complementary to the 5' end and the other complementary to the 3' end. The DNA to be cloned is put in a tube with DNA polymerase and the cofactor magnesium ion and 4-deoxyribonucleoside triphosphates, DNTPs. The primers anneal to the denatured DNA, and the complementary strands are synthesized by heat-stable DNA polymerase. The three steps of PCR are denaturation, primer annealing, which is hybridization and annealing, and extension. The steps are repeated over and over, using a thermal cycler to amplify the DNA exponentially. The DNA strand is doubled in each cycle. New strands along with the old strands serve as templates in the next cycle. In PCR, the target DNA is denatured into single strands. Each strand is then annealed to short complementary primers. DNA polymerase extends the primers in the 5' to 3' direction using the single-stranded DNA as a template. The result after one round of replication is a doubling of DNA molecules to create two newly synthesized double-stranded DNA molecules. Repeated cycles of PCR can quickly amplify the original DNA sequence more than 1,000,000-fold. Please note that when you look at this slide shown here, it's a relatively short sequence of DNA being amplified. Typically, much longer segments of DNA are used for PCR, and the primers bind somewhere within the DNA molecule and not so close to the end of the actual molecule. So in looking at applications of PCR, it's a useful tool, most widely used technique in genetics and molecular biology. It allows for screening of mutations involved in genetic disorders. The location and the nature of mutation can be determined quickly utilizing this method. And allele-specific probes for genetic testing can be synthesized, making PCR important for diagnosing genetic disorders. However, there are some limitations of the PCR method. Some information about the nucleotide sequence of the target DNA is required to synthesize the primer. Minor contamination from other sources, including skin cells from a researcher, can cause problems by contamination. And PCR cannot amplify long segments of DNA. Please view this video on PCR. We'll now look at southern blotting, which is a method used in molecular biology for detection of a specific DNA sequence in the DNA samples. Southern blotting combines transfer of electrophoresis-separated DNA fragments to a filter membrane and subsequent fragment detection by probe hybridization. Southern blotting was named after Edward M. Southern, who developed this procedure at Edinburgh University in the 1970s. The Southern blot determines whether the clone contains all or part of a gene. It ascertains size and sequence organization of gene or DNA sequence of interest. It has two components: separation of DNA fragments by gel electrophoresis and hybridization by using labeled probes. In the Southern blotting technique, samples of the DNA to be probed are cut with restriction enzymes, and fragments are separated by gel electrophoresis. The pattern of fragments is visualized and photographed under ultraviolet illumination. The gel is placed in an alkaline solution to denature the DNA into single strands. Then it is placed on a sponge wick that is in contact with a buffer solution and covered with a DNA binding membrane. Layers of paper towels or blotting paper are placed on top of the membrane and held in place with a weight. Capillary action draws the buffer through the gel, transferring the DNA fragments from the gel to the membrane. Single-stranded DNA fragments on the membrane are hybridized into a labeled DNA probe. The membrane is washed to remove excess probe and overlaid with a piece of X-ray film for autoradiography or chemical luminescence from the probe detected with a digital camera. The hybridized fragments show up as bands on the X-ray film. If we look at the slide and we look at A, the agarose gel is stained with ethenium bromide to show DNA fragments. In B, exposed X-ray film of a southern blot prepared from the gel in part A shows you that only those bands containing the DNA sequences complementary to the probe show hybridization. Southern blotting led to the development of other blotting techniques, including northern blotting, which is RNA blotting, and western blotting, which is used to analyze proteins. Please go to the video to watch a video on southern blotting. Fluorescent in situ hybridization fish, we've talked about before. It involves hybridizing a probe directly to chromosome or RNA without blotting. It's carried out with isolated chromosomes on a slide or in situ in tissue sections or entire organisms. It's helpful when embryos are used for various studies in developmental genetics. And you can go to this video to see the technique of fish. If you look at individual genome sequencing, we see that it's utilized in medical clinics. It's changing the ways that scientists and physicians evaluate a person's genetic information. It provides insight into the genetics of Alzheimer's disease, Proteus syndrome, anorexia, and many other diseases.